So to better understand why cells like to perform glycolysis and how the glycolytic pathway can be self-sustained, the questions which we posed in the previous part, it is important to bring glycolytic pathway into the context of whole cell metabolism. If we take intermediates in the glycolytic pathway, it is there is almost no single intermediate which is not brick in some other biochemical pathway important for synthesis of important biomolecules. For example, glucose 6-phosphate is an origin of pentose phosphate pathway, which is then necessary for synthesis of ribose and deoxyribose, which in turn give rise to nucleotides, important, essential for flux of information, biological information in all the cells. Likewise, fructose 6-phosphate is important intermediate in amino sugar biosynthesis, and then this is important for synthesis of various glycoconjugates, which we discussed when we talked about carbohydrates. Glyceraldehyde 3-phosphate is at the end used to synthesize phospholipids essential for cell making cell membranes, for example. The 3-phosphoglycerate is undergoing simple transamination reaction to produce amino acid serine. Phosphoenol pyruvate, being unsaturated compound, is important precursor for synthesis of aromatic amino acids, phenylalanine, tryptophan, and tyrosine. The pyruvate, in addition to its role of as an intermediate for synthesis of acetyl-CoA, which in turn is burned in mitochondria in the oxidative phosphorylation to produce ATP. The pyruvate is also a precursor of a number of different important biomolecules. So it is, for example, precursor in biosynthesis of a number of amino acids like alanine, asparagine, or aspartate, and others. It's also carboxylated to produce oxaloacetate, and again, oxaloacetate is an important precursors for various pathways, one of which, for example, gluconeogenesis, which we are going to discuss in the next part of this lecture. So altogether, it is obvious that the production of energy in the form of ATP and NADH reducing equivalence in the form of NADH is a product of glycolytic pathway, and besides that, it is centrally embedded into the whole framework of cellular metabolism. So this answers kind of part of the question why cells, why glycolytic pathway is so important for cells. So, but to understand how this glycolytic pathway could run, so we also need to understand how in our cells it could be fed. And the situation in our organism, in our cells, is much more complex than the just starting from glucose. The simplest case scenario in our cells is that the glucose is fed into cells in the blood, in the form of blood supply. So, and in this case, the glucose has to be engulfed within the cell, and this happens through the action of glucose transport channels, which going to be discussed at a later point. So once it is getting into the cell through the these GLUT channels. It is phosphorylated and then goes through this glycolytic pathway, as we discussed in the previous parts. So another way how it could be fed is through intracellular sources. When we talked about carbohydrates, we discussed the pivotal role of the glycogen in our cells is a source of glucose. So it could be converted to glucose very quickly by the phosphorylation with inorganic phosphate phospholysis using enzyme glycogen phosphorylase, and then after being converted to glucose 1-phosphate, not confused with glucose 6-phosphate. It is different isomeric form. Sorry, but this one can be converted with the action of phosphoglucomutase to normal glucose 6-phosphate and then run through the pathway. But there are also more complex situations. So we get a lot of different kind of carbohydrates, different kind of sugars, polysaccharides and oligosaccharides in the form of dietary supply. So at the end, they need to be somehow processed, engulfed and processed in our intestine to get transferred to our cells for subsequent subsequent feeding the glycolytic pathway. And there is a complex system of enzymes which can feed at the end glucose, convert everything into glucose and feed it into glycolytic pathway. For example, so there are a number of sugars which could act as a dietary supplies, like for example lactose, triclose, or sucrose, the respective lactic sugar, cane sugar. Yeah, and so in these ones, these are disaccharides, and they are cleaved in our intestine by glycolytic enzymes, lactase, triclase, and sucrase, respectively. So, this is the point where the products monosaccharides can be engulfed in the cells. Disaccharides cannot penetrate through cell membrane, and for example, there is a condition in many people, lactose intolerance, which is due to the fact that the low expression level of lactase in some cases, so the lactose cannot be cleaved into these monosaccharides, constituting monosaccharides, glucose and galactose in a small intestine, and then it goes the lactate goes through lactose goes through lower intestine where it is consumed by the bacteria. So after cleaving into these monosaccharides, some of these products actually consist of pure glucose, and like for example the dietary glycogen or starch, they are cleaved by enzyme called alpha amylase, and then actually it is pure glucose, so it can be and go into the cells and undergo normal glycolytic pathways. But some of the monosaccharides are not glucose, but some different sugars, and then there is the complex intracellular system, usually a system of enzymes which converts these sugars into form compatible for running the glycolytic pathway. But the common theme among these feeding enzymatic pathways is that the monosaccharides getting into the cell first fixed inside the cell by usually by phosphorylation or uridinylation. So in this way, these sugars cannot get out of the cell back, so they are fixed in the cytosol. So in some cases, these are phosphorylation events. For example, the like glucose, the fructose, which is getting in the cell, or mannose, which is epimer glucose, are all phosphorylated by action of hexokinase, one enzyme. But for example, galactose is urinylated, converted to the UDP form. And then, well, some of the sugars would need to undergo stereoisomerization to get into this comparable form, like galactose and mannose. At some point, they get isomerized by respective isomerase and fit into the different parts of the glycolytic pathway, in a sense. Yeah, and so also there are different ways how the sugars can enter. They can enter the glycolytic pathway at a different level. So fructose, for example, it can enter the glycolytic pathway two ways. Once through this hexokinase mediated phosphorylation, and another through fructokinase mediated phosphorylation, which allow to bring respective intermediates into different parts of the glycolytic pathway. But altogether, we could see that there is a very complex system toolbox in our cells, which allow to feed various carbohydrates into the glycolytic pathway. So this allows it then to burn the carbohydrates in glycolysis. So another important question which we posed but didn't cover is how the glycolytic pathway could be self-sustained. In order for glycolytic pathway to proceed forward, the NAD plus, the electron transport carrier, needs to be reduced to NADH. In aerobic conditions, this is not a problem to replenish the source of NAD plus by feeding this NADH one or the other way into electron transport chain and burning these electrons, transferring them to oxygen. However, in anaerobic conditions, this is a problem, and to sustain the glycolytic reaction and glycolysis in the anaerobic purely fermentative mode, there is enzyme lactate dehydrogenase, which takes pyruvate as a substrate and then reduces pyruvate to lactate using NADH, and this way replenishes the source of the NAD plus. So, and this is called lactate fermentation. So, and altogether, if we take this anaerobic lactate fermentation mode of glycolysis, which takes place in fact in some of our cells, as we will discuss later, the glucose in the initial point substrate, and then through this series glycolytic reaction and one extension, the glucose is converted to lactate molecules. And if we look at this initial product and final product closer, we would see that there is no reduction of or oxidation occurred after all. So this total reaction and production of ATP through lactic acid fermentation is driven purely by complex rearrangement of chemical bonds within the glucose molecule, which at the end it produces 2 ATPs and actually produces a lot of free energy in the form of heat. So in an essence, this is from the point of ATP production, it would be very inefficient energy draining step given low yield of ATP. Nevertheless, it is taking place in some cases. One obvious case are anaerobic conditions when there is no enough oxygen supply, and this is the case, for example, in our muscles during the intense workout, where there is simply not enough oxygen supply to produce ATP to run muscle contraction. In these cases, muscle cells burn very quickly the internal glycogen supply, convert to glucose, and produce ATP through lactate fermentation. And this is one of the reasons why the after hard workout next day there is a muscle strain. This is because of lactate buildup in muscle, which is not evacuated in a timely manner. Another case are obvious cases like erythrocytes, which is a case of actually aerobic lactic acid fermentation, because erythrocytes simply do not have mitochondria to burn ATP through oxidative, to burn glucose through the oxidative phosphorylation. Of course, some microorganisms prefer to do lactic acid fermentation, like lactobacteria. So there are adverse cases where the lactic acid fermentation takes place, and this is common in various kind of cancers, both aerobic and anaerobic. The anaerobic scenario usually takes place just because cancer cells, tumors do not have enough oxygen supply through blood vessels, but it's commonly known that many tumors perform lactic acid fermentation even at aerobic conditions, which is known as Warburg effect. And there is a reason to believe that this is a common scenario because of the fact that glycolytic pathway produces not only ATP but a lot of precursors for various biomolecules, which is practical for quick building of biomass for proliferation of cancer cells. But another mode of regeneration of NADH and for cell-sustaining glycolysis is this two reaction of ethanol fermentation, without which probably students' life wouldn't be as fun. So in ethanol fermentation, there are two enzymes, pyruvate decarboxylase and alcohol dehydrogenase, which respectively first remove this carboxylic group from pyruvate in the form of CO2, producing acetaldehyde, and acetaldehyde is then used as an electron acceptor from NADH to regenerate back NAD plus to feed it back into glycolysis and produce ethanol. So that's what yeast would like to do in anaerobic environment. So the interesting enzyme here is pyruvate decarboxylase. It has this cofactor thiamine pyrophosphate, which acts as a coenzyme, and essentially it acts as a prosthetic group which kind of tightly embedded in the context of pyruvate decarboxylase. And this case is interesting in the sense that thiamine pyrophosphate is a cofactor coenzyme in variety of different chemical reaction. In our organisms actually needed as a dietary supply known as vitamin B1. So there's plenty of it in yeast because of ethanol fermentation. So, and thiamine pyrophosphate has this important group thiazolium ring, which could serve as kind of active aldehyde acceptor in the case of ethanol fermentation. Uh, active aldehyde carrier, and in yeast ethanol fermentation it serves as active acetaldehyde carrier. So the aldehyde group is bound in this some kind of chemiacetal form, and this is useful in various scenarios. In the case of pyruvate decarboxylation, the this the active aldehyde bound form of thiamine pyrophosphate is simply rearranged and falls apart back into thiamine pyrophosphate and the acetaldehyde. But in the case of other metabolic pathways, metabolic reactions, for example, in the case of pyruvate dehydrogenase, producing the acetyl CoA, for example, from the pyruvate. In this case, this intermediate step that finally leads to production of acetyl CoA. This group is again is transferred to coenzyme A. In the case of transcatalase, the thiamine pyrophosphate acts as important carrier to stitch and split the sugars